I'm very happy to be here, and hopefully all of you can see the screen and hear me clearly. So today I'm going to talk about how we can use genomics, more precisely evolutionary genomics, for biodiversity and conservation. This is a topic that I've been studying since my postdoc, and hopefully I'll be able to persuade some of you that both from a perspective of evolutionary biology and from the perspective of understanding biodiversity and conservation, using genomics is very important and very fun and very useful. So I'll just begin with my classical opening about the genomics revolution, which basically genomics really has revolutionized the fields of medicine and agriculture. And I emphasize these two fields because these are the two fields which genomics has impacted the most. And these are the two fields where genomics has, or to say it another way, these are the two fields which has led to both the genomic revolution and to most of the methods that we use in genomics. Like most of the bioinformatics methods we see today are directly related to understanding human health and also understanding agricultural yield. So what do we mean by this? As you know, one of the core aims or core objectives we have today is understanding personalized medicine. And this has a lot to do with understanding the genomic architecture of diseases, genomic architecture of viruses, how our bodies work, and how we respond to all sorts of influences, and how our genomes respond and process all of that information, and how we can actually understand how we can go from genomes to phenotypes to actually understand all of the processes that might harm us and we need for our health. So again, medicine, the field of medical genomics has been very influential in both using genomic data and also developing certain tools to use to analyze and understand the workings of our genome. Likewise, genomics has also revolutionized the field of livestock or agriculture because a huge part of one of the biggest challenges we have today is, of course, feeding our ever-growing populations, and we need to have higher. We need to produce higher yields. If we're talking about, agriculture, we need higher yields. If we're talking about livestock, we need, livestock that produces more milk, gives higher milk yields, and genetics is very good at that. In the old days, as you know, we did crosses, breeding programs, which took decades. You have to cross the right breed. You only have morphology to go with. Today, we can do genomic predictions. We can just take, for example, a seed, look at its genome and predict what type of yield it might give. And so we can, take two seeds. If we know their genomes, we can predict what that cross will produce. It's the same thing for animals as well. Again, this is one of the main topics, like genomic predictions, that are used heavily in agriculture, and which is, a huge part of the agricultural revolution that we're going through. And so what I'm trying to say here is that genomics has revolutionized both of these fields, and most of the tools that we use in bioinformatics actually directly comes from these two fields, because that's where the emphasis has been. What has not really been emphasized much in this genomics revolution is, of course, environmental science, and it has been relatively neglected. So I don't mean that genomics is not being used in the science in environmental sciences or conservation biology. It is, and we'll get to that, but it's not at the same level as we see in agricultural and the medical sciences field. And neither is there as much incentive in these fields to use the entirety of genomic information in answering questions about environmental science or conservation. I'll try to explain what I'm saying by that. And neither, when we're saying, it's not as important, it is as important, because I always give this example. For some reason, environmental science and conservation is always neglected when we compare them to agriculture and medicine. But I always give this example. We can reach a stage in our knowledge and our technology that we can cure any diseases that every single human born could live to their maximum genetic capacity without going through any diseases or dying prematurely because of some genetic factor. So we could solve all of that. We can have the perfect personalized medical cures, and so everyone can live up to their maximum genetic capacity. We could achieve this. That is a goal. It's a lofty goal, but that can be achieved. We could also achieve like huge yields in agriculture so that we can feed billions of people in the world and no one would actually have food shortage. We can even today with our technology achieve such things. But it doesn't really matter if we can live as much as possible or if we can just like feed as many people as possible. None of that really is important unless we have a proper place to live. And that's where conservation biology and environmental science comes in, because in order for that to have any meaning, we need our habitats, we need a home to be able to live in. And up to this day, we only have this one single globe that we live in. We have just one world, and if we don't take good care of it, it doesn't really matter how good our technology is. It doesn't really matter how good we are at curing people. It doesn't really matter how good we are feeding people. If we don't have a place to live, none of this actually matters. And that's why conservation biology, environmental science, and the Conservation Act, the Green Act, should be getting as much emphasis and as much scientific grants and as much given as much scientific importance as these other fields. And happily we're getting there. It's not as good as we want it to be, but we're slowly getting where both institutions and organizations are understanding the importance of conservation and understanding the importance of using as many of our knowledge to actually help save our Earth or help protect our Earth so it can last longer. So let's get to the role of genomics. As I said, it's like when we mean relatively neglected, we're not saying that it's never been used, but it's usually been used in that type of way. So mostly what we do about whether this, like we started with, one or two markers, today we can do whole genome sequences, but mostly what genetic data is used in environmental sciences is to understand patterns or global patterns of genetic diversity. So the typical study would be to, if we have an organism, like it could be several species, it could be one population that is distributed across a certain landscape, what we can actually do is we can understand whole genome diversity of this organism, of this species, or of these several species, doesn't matter, along this landscape, and figure out in which regions we have low genetic diversity, in which regions we have high genetic diversity. And this is pretty good proxy for understanding which organisms, like which populations we should emphasize or which populations we should actually protect, because low diversity populations are populations which are much more vulnerable to environmental change. So perhaps scanning for these general patterns of diversity is always helpful in understanding which populations might need our help or which populations might be under threat of extinction. And this is usually the framework where genetics is mostly used in environmental sciences or for conservation purposes, just understanding patterns of genetic variation. And sometimes this is very effective. So when pattern actually equals processes, so when overall patterns of genetic diversity also tell us something about the process of how that genetic diversity arose, then it really is a good proxy for answering conservation questions or for determining conservation units. So here's a study that we did in 2017. And I'm not going to go to the details, but this is a good example. For example, here the question before we started the study was that, so we have one fish species that is distributed around the western coast of the United States. And the question was, well, is this black population genetically distinct from the others? Because there were some indications that it might be. And if it is, do we need different conservation measures to protect this population here relative to these populations here? And when we actually did the work, so this is genome-wide genetic diversity, we actually very clearly see that it is genetically highly differentiated. Its diversity patterns is very different from all of the other populations within this basin or within these basins. And so from this perspective, we can say, yes, genetic diversity is very different. This is genetically a totally different animal. So yeah, if we're going to implement some conservation measures, the conservation measures that we implement for these aren't necessarily the same for this one. So again, in this sense, using large-scale genomic diversity patterns can help us, can inform us about how we should go about protecting species. But this is not always the case. So here's another example, and a much more interesting example of a study that came out in 2020. So here what we're looking at is, again, the basic question was the same. Here we have a fish species, the name of the species is not really important, which is distributed along the western coast of the United States. And the question here was, okay, here we have San Francisco, and these are other natural lake systems or basins within the western coast of the United States. The question was San Francisco. Of course, San Francisco is a huge city. It's a huge commercial hub, and there's lots of disturbances in the Bay Area. So the question was, are fish, populations of this species within the Bay Area, are they under threat? What is the diversity level? Are they under threat of extinction? What is their effective population size? Is it going down? And should we take measures to protect these fish in the San Francisco Bay Area? How much isolated are they from the same species, or populations of the same species in other more natural and less disturbed habitats? So again, when we look at genome level diversity, genome-wide diversity levels, we can actually see that the Bay Area populations are doing pretty fine. They have high levels of genetic diversity, and they're not really differentiated from other nearby populations. And so this would suggest that there's no real reason to panic about what's going to happen to these fish populations in the Bay Area, even if they do get extinct, even if they're under very high disturbances. Genetically, they are highly diverse, so they can buffer a lot of things. And they're also very similar genetically to nearby other populations in the basin. So even if this population went extinct, we would still be protecting much of the genetic diversity in other pools. And so we could very easily recolonize this population again if we wanted to. So if we look at general patterns of diversity, we don't actually see any reason to go ahead and implement strict management plans in this area. But if we actually look at the processes, not just the genetic patterns, but if we look at the processes that has resulted in the observed patterns of genetic diversity, now this actually tells us a very different story. So just very shortly, so one thing that we learned when we look at the processes, both the ecological and evolutionary processes that resulted in these patterns, we can actually see very strong signals that there is forward migration. So there's migration from the Bay Area to nearby populations, but we don't actually have migration in the reverse direction. So there's migration from Bay Area populations to these other populations, but none of these populations, we don't see actually migration from these populations back into the Bay Area. So that's very important. Another thing that was really important in this study is that we also see loci or genetic diversity that is adaptive in these populations. And we know that those adaptive loci originated from the Bay Area. So what is this telling us? This is telling us that yes, Bay Area has high genetic diversity, but this is the source population that sources out all of its genetic diversity to other populations. So it feeds other populations, and it's also the source of adaptive divergence in these other populations. So this is very important. So this makes the Bay Area population a very important population to protect, because it is the single population which actually feeds all of the rest. So if we actually lost this population, we would lose, these populations would lose a source of genetic diversity and a source of adaptive variation. So again, this shows us that it's, sometimes just looking at general patterns of diversity and making the decision is not enough. And we actually have to look at the processes that have brought about that genetic diversity. So again, in this case, if patterns don't always reflect the process of divergence, then understanding divergence itself, understanding adaptive and ecological processes that have produced that variation becomes very important. And that can really change how we approach conservation measures and how we approach protecting these populations. Okay, and this all leads to the important, again, so this is not really important. Again, just to summarize what we've said, this shows that evolutionary genomics is a very important tool for understanding history, process, and function. It can give us information about all of these. So we don't just have to use genomics to understand or describe patterns of diversity. We can use evolutionary genomics because we have genome-wide information to understand the history, processing, functions that are going on. And that can, of course, reflect back onto how we implement protective measures or conservation measures. And this leads to the not the new field, but the slowly emerging field of applied evolutionary biology. So applied evolutionary biology is still very it's not something very popular, or it's not something well known. But again, evolutionary genomics is actually a very applied field, or it should be an applied field, because today we know that most of the global problems that we see around that we're facing, for example, the food crisis, the health crises like chronic diseases, the environmental crisis, most of these actually have to do with evolutionary biology. What do we mean by that? When we look at most of our global problems, let's say we have emerging chronic diseases where already, like, our health is deteriorating. We're seeing many more diseases that we did before in the past. And most of this can be traced back to the mismatch between the phenotype and the environment. What do we mean by this? We as humans have been adapting to our environment for over, if we look at modern humans, that's about 250,000 years, give or take. If we take that from the split with the chimpanzee, that's about 7 million years. We've been adapting to a certain type of environment. And for a very, very long time in our history, let's say until the agricultural revolution, roughly around 6,000 years ago, we were mostly hunter-gatherers. So we fed on what we found. Finding high sugar foods was nearly impossible. We gathered seeds, we ate some insects. If we could hunt a game once in a while, we could eat meat, etc. So our diet was very specific. And we've been adapting to that diet for a very long time. But right now, all of a sudden, our environment doesn't really reflect our adaptive history. Our environment now has, we have access to a lot of carbohydrates. We have access to a lot of sugars. We have access to fatty foods whenever we actually want. So there's a mismatch between what our bodies have adapted to and what our environment is giving us. And this causes a lot of problems because our fitness is not, our fitness maximum isn't to this environment. It's an environment where, carbohydrates are limited, sugar is limited, fatty foods are limited. So that's the type of environment which we're adapted to. But right now, our environment doesn't reflect that. And with that comes a lot of chronic diseases. The way to fix this is to move our adaptive landscape into our new environment. So we need to, kind of shift our evolution towards the new environments that we're living in. So this, again, can only be done if we understand evolutionary biology, if we understand evolutionary genomics, if we understand the mechanism of change, how organisms respond to environmental changes, and if we understand the genomic architecture of this, then we can start slowly building models that will enable us to move this fitness, our current fitness, into the fitness that is necessary to deal with most of these problems. Again, you can say the same thing for biodiversity. The environment is changing very fast, and most organisms are now have a phenotypic mismatch with their new environment. They're not used to living in these modern environments, in these modern urban environments, which are changing very quickly. And a lot of organisms are having problems adapting to that. But if we understand adaptation, if we understand how they're adapting to environmental change, then we can use this to shift their fitness towards the environment that they're in. And that's the framework that I want to give you today, because most of conservation biology concentrates on protecting natural habitats. And what I'm trying to emphasize, and our lab group is trying to emphasize, is that I think conservation would be much better served if we try to emphasize adaptation to changing environments and not to old environments. Because after a time, environments are always going to change. Instead of restricting animals to their quote unquote natural habitats, I think it's more important to look at conservation as how can we conserve species in a changing landscape? What do we have to do? What type of information do we need to enable protection of species in changing landscapes? And that's where adaptation comes from. And that's where the evolutionary basis of adaptation is very important, because if we understand the evolutionary basis of adaptation, then we can move our protection or our management strategies towards protecting that adaptive variation that will enable the organism to survive under changing environments. And I'll give two examples about these types of studies. So the first example I'm going to give from the study we did in the United States together with Mike Miller and Dan J. Priss. Dan was our PhD student at the time, so of course he did like 90% of this work was done by him. And so this is a great topic to discuss this framework that we're talking about, and we're going to see how understanding evolutionary basis of adaptation of salmon can actually inform our conservation decision. So salmon, of course, very iconic fish. They spawn in the rivers, and then they migrate to the ocean, and they spend some time in the ocean, mature, and after they've matured, then they migrate back to the rivers to spawn and to reproduce. And of course, it's fascinating because what they actually do is once they spend a lot of time in the ocean, they can somehow hone in back to the exact position which were they born in. So again, that's also very interesting. So they're very iconic species. They're also very iconic because in the summer months you can actually see them, or in the spring months, summer months, you can actually see them like climbing up these waterfalls, these fast flowing waters to reach the upper lakes which they were born in. But there is one problem with this. Again, so what salmon actually do is they migrate, they make this migration after maturity in spring, summer months. But as global warming increases, so as the globe starts to warm, what the fish need is they actually need very cool waters. They need that cool, fast running waterfalls. And when they reach their birthplace, they need also very cool habitats, cool waters to be able to reproduce properly. So that's the environment which they're adapted to. But since global warming, and since they make this migration towards, in the end of spring and summer, as temperatures warm, the flux of the water and the flow of the water is reduced. Waters are getting hotter, so it's been much more problematic for these fish to make that journey back home. And a lot of fish can't actually make it as well because they need those cool running waters, both to be able to make that migration and also to be able to reproduce once they're in those waters. But if those waters are hot and if they're not really flowing as fast, they can't really make that journey. So again, that's a huge problem for salmon fish, and it's a very sad result of global warming. But there is a bright side because there is adaptation going on in these fish as well. So some of these fish, instead of maturing and making migration, they're actually doing a behavior which is called premature migration. So what's premature migration mean? So migration is usually in the spring and summer, but what premature migrants are doing, instead of making that migration in the spring and summer, they're actually making that migration in the fall. So again, when when environments are much more cooler, so waters are much more cooler, and warmth has not set in. So instead of waiting to spring and summer, which is now not ideal for their migratory behavior, they're actually doing this much more early. But this comes at a cost. This comes at the cost that right now they're making, beforehand they're making this migration after they've matured, but right now they're doing this migration before prematurity. So what does this mean? They actually have to make that migration from the ocean into their rivers and to the lakes, but then they have to mature in the lakes. And again, the lakes are much more restricted environments. Oceans are vast. It can have much more higher fish. You can find much more higher yields of food. There's also food sources. But once you're in a small lake or a small river with lots of fish, then maturing, finding enough resources to mature is going to be problematic. Again, so that's the adaptive part of it. Again, the trade-off is, yes, you can use those cool running waters to get back to the region where you're going to reproduce. But now you have the problem of maturing in a much more resource limited environment. But these fish, the premature migrants, solve this by having faster developmental rates and faster rates of fat yield. So they can actually mature much more faster than premature migrants can mature much more faster and using much less resources than the mature form. And when you look at overall genetic diversity patterns, so here we have, as you can see, in lots of different lakes and lots of different areas, you can find both the mature and the premature form. So the triangle is the premature and the circle is the mature form. And when you look at genetic diversity, they're not really different from each other. So these two life history variants, overall genetic diversity is very similar. As you can see, mature and premature migrants from this orange area are genetically very similar to each other, and they're very distant from, for example, mature and premature from the blue area. So this means that this variant is not really closer. So it's geography that determines genetic diversity, and this means that since these fish are genetically very similar, each population is very similar. This means that genetically, probably this variation is very abundant in the population and can naturalize multiple times. So that's one thing. So one hypothesis is that, since we spot this variation in a lot of different independent populations or locations, then this means that this is a variation that can very much readily evolve. And this has important consequences for conservation, because this means that we don't actually have to conserve this adaptive variation by itself. We can just concentrate on conserving, a few populations. But what we want to do answer was like, what can evolutionary genomics, like, let's understand the architecture and the evolutionary basis of this this shift in life history, so to speak. And when we looked at it, we actually saw that most, the genes that actually explained this variation was mutations in this GRB1L gene. So mutations in the GRB1L gene made the switch from a mature migrant to a premature migrant. And GRB1L gene is a metabolic gene. It's also a gene that is linked with childhood obesity in humans. So again, it's a metabolic regulatory gene, and what it actually regulates is that it regulates it quickens, speeds up development, and it also enables much more higher fat storage from development. And this makes sense. Again, this makes sense in an environment where it's resource limited and you have to grow very fast and you have to grow to large sizes in very resource limited environments. So again, mutations in this GRB1L gene can actually tell us about the differences between premature and mature life forms. But what was most interesting is when we look at divergence within this gene, so how this gene diverged for evolutionary history, we can actually see that no matter which geographic location a premature migrant is coming from, in this gene region, they're very genetically similar, and so are the matures genetically very similar. So this tells us that this variation didn't evolve independently multiple times. It actually evolved once and then spread. So this means that this is not a very common variant. This is a very single allelic event. This is a very important singular evolutionary event, which must be protected, because if we lose this variant, it's very highly unlikely that we're going to see this adaptation again. So this changes the focus of, instead of protecting populations, maybe we should just try to protect this very important adaptive variation, which will enable these species to survive in changing environments. And again, of course, in the United States, this thing is a bit more easier. You can get a lot of press, but again, getting from pure science to actually applied changes is very hard, and it's still again, there's always a huge difficulty. So the question is, can we just look at can we just conclude that if we know genetic mechanism of certain change, would that be enough to change our listing of endangered species, or how we do conservation? So this is an continuing discussion. So the next example I'm going to give is from home, here from Turkey. And again, this is a topic with which we've been working on for the past 2 years, roughly 2-3 years. And that's adaptation of brown bears to human-dominated landscapes. And we're doing this study together with Can from Can Şekercioğlu from Utah University, and our PhD student, Kemal Aytekin, she's here. And of course, like, most of the work, again, is done by Çisel. So most of the results that I'll be summarizing is it's due to the hard work that Çisel has put into this project. So let's give a little background information about Eastern Turkish brown bears. Now, when we look at Eastern Turkey, the availability of human resources, when we look at it, there's a very large population of brown bears, especially Eastern Turkey, mostly around Turkey as well. But when we look at it, they don't really have much natural resources. So again, their natural prey and their natural food sources are actually diminishing in these areas. But contrast to that, they also have a lot of the availability of human resources is very high in these areas. And this has led to the observation, a shift in feeding behavior, because what we actually see in this area are two types of bears. We have wild foraging or migratory bears, which usually breed around or spend winter around Sarıkamış, but then they actually do this, they migrate all the way up to sometimes, in this case, towards Artvin, sometimes into Georgia, and sometimes into other areas and search for food, and then they come back to hibernate. Whereas contrast to that, we have other bears, which we've named quote unquote sedentary bears, which have actually established themselves in the Sarıkamış garbage dump here. So Sarıkamış is an open garbage dump, and these bears, which are sedentary, actually just spend all year round, like roughly around 1 or 2 kilometers around this garbage dump. They feed solely on the resources that are within the dump, and they don't actually go forage for food, as opposed to migratory bears, which like forage, several kilometers in search for food and then come back to hibernate. Again, so there's this very interesting life history shift within these bears, where we do have some bears that are very sedentary, spend all of their time and feed exclusively on the garbage dump. And we have these migratory bears which don't actually feed on the garbage. They don't use the garbage dump as a food source, but they forage for their own food. And what we were interested in is, okay, so can we understand the genetic basis of this life history shift and what that can tell us about how to conserve these species? Because the important thing is, if this is a learned behavior, this can be, this could be discouraged, for example, then just feeding off the humus hocus can be discouraged. But if this is an adaptive feature, if we actually have a genetic basis for this shift, if we're really having an evolutionary change, we're observing evolutionary change, then that is a different topic of discussions. Again, the topic comes to, well, if these bears are adapting genetically to their new urban environments, do we have the right to go ahead and restrict that evolution back into their quote-unquote natural habitats, which are always going to be restricted, because humans are not going anywhere, our environment is not going anywhere. So the debate is, if we have adaptation to urban environments, and if urban environments are increasing, and if these bears are being forced to live in these environments, should our conservation measures prevent them from adapting to these new environments? So that's the question that we were trying to answer in this study. And let's just start very quickly with, and I'll go through these very quickly. So first we looked at overall genetic patterns of diversity, and these were very interesting in themselves as well. So here we contrasted them with genome-wide patterns in Apennine bears. So Apennine bears are Italian bears which are trapped on the high mountain peak of the Apennine Mountains. They've been isolated for thousands of years, so they're very genetically distinct. They're a very small population, like effective population size is very small. There's huge amount of genetic drift, so they're genetically very, very low in genetic diversity, and they're very distinct from European bears. So here we see European bears, here we see bears from Alaska, and here we see bears from Russia. But the most interesting part is that the Eastern Turkish bears are actually as much different from European bears as nearly these Apennine bears. So from a glance, from just overall genetic diversity patterns, we actually know that Turkish brown bears are very different from European brown bears, almost as genetically distinct as this very, very small population that has been isolated for 1,000 years. That in itself is very interesting, because there's no reason that Turkish brown bears should be that isolated. Another interesting result was that even though that we're highly isolated from European bears, we actually harbor much higher genetic diversity than nearly all of the other bear populations. So you can actually see here, green is the Apennine bears. This reddish orange thing is the Alaskan bears. Here we have this lilac color is the European bears, and this colorful picture here are bears from Turkey. So what we're actually seeing is we're genetically very different, highly very different from European brown bears, but we're also genetically very diverse. We have much more, mixed ancestry and much higher diversity than European bears. So this in itself was interesting. We're talking about a small Eastern brown bear population, which is very different, but also harbors a lot of genetic diversity. But when we actually look at whether genetic diversity pattern differs between sedentary and migratory bears, we don't actually see a difference. So genetically, overall when we look at genome-wide patterns of diversity, they're very similar. So brown, we can't really differentiate based on genetic diversity patterns like migratory and sedentary forms. So what we next did was, are there genes that are responsible? So can we find genes that can separate these two life histories? So we did a genome-wide selection scans or genome-wide association studies, and then we looked at signatures of positive selection in our top 20 associated SNPs. So of the SNPs that were associated with this life history variation between migratory and sedentary bears, we looked at the top 20 SNPs, and we looked at whether we saw signatures of positive selection, meaning from these 20 SNPs, are there some SNPs that are highly differentiated in sedentary bears as opposed to migratory bears and European bears? And are there some SNPs or genetic regions that are very differentiated in migratory bears as in relation to sedentary and European bears? And our criteria for significance was very high, so we chose only our error rate is 99.9%. So this is a PBS analysis looking at extreme differentiation, and only extreme differentiation over 99.9% of genes that were different is the regions which we deemed significant. And for migratory behavior, we found 1, 2, 3, 4, 5 genes, or 5 genomic regions that showed signs of high differentiation from migratory bears, and we found another 4 in sedentary bears that were highly differentiated from migratory bears. And when we looked at these, most of these, some of them didn't really have any gene regions next to them. So we looked at 1 kb, approximately 1,000 base pairs. So we looked at the genes that lie 1,000 base pairs along these SNPs. And most of the genes were transcription factors, which made sense. So modifying transcription events. But one of them here, as you can see, which was actually the CCRL2 gene, which is a very interesting gene, because here the difference is that we actually so one of the SNPs were actually right in the exonic region, in the coding region of this gene here. And what we actually notice is this is the gene that controls immune response. So we were actually seeing, when we looked at migratory bears, we were actually seeing a selection for immune response. So we were seeing selection in a gene that had to do with immune response, and this actually made sense. Again, the immune response of bears that feed off of natural human resources, as opposed to having more of a migratory behavior or actually having wild foraging, this could be very important for their immune response. So that we did have a very important hit showing that immune response of these 2 migratory bears was under evolutionary pressures. So we wanted to see things further. So this was a classical association study. We actually wanted to do a polygenic discrimination. So this meaning that sedentary and migratory behavior is very complex. So there's no reason that we should believe that there are 1 or 2 genes that are responsible for these types of differences. So what we want to do is we want to model polygenic selection. We want to model the polygenic architecture of this differentiation. So all of the genomic regions that could be playing a role in the differences that we see in sedentary and migratory behavior. And what we actually found was we found about 464 SNPs that were contributing to differences in sedentary and migratory behavior along the genome, contributing to foraging behavior. And among these 464 SNPs, we actually found 514 gene regions that were, that played a role in this differentiation. And then we looked at gene ontology and to see which pathways was, regulated, which pathways was heavily regulated within these 514 genes. And as you can see, most of the genes that were heavily regulated or upregulated from the switch from sedentary to migratory behavior, or that explain the difference between migratory and sedentary forms, were actually transcription factors. So this again makes sense. We're actually not seeing too much structural changes, but we're seeing changes in regulatory regions which actually regulate behavior. But most of the genes were actually those genes that regulate sensory perception and smell. So again, this is very important for foraging behavior. Again, we don't actually know the functional importance of these genes, so that's going to be the next step of our study. But we do know that most of the genes that are influencing behavioral changes have to do with sensory perception and smell, which makes sense because Again, bears that use the garbage dumps and human resources heavily must actually tolerate, different smells than, going around and foraging food in wild environments. So we're actually seeing an adaptation towards sensory perception in smell when we look at Go terms. And when we also do pathway analysis, we get the same results. The genes that are heavily regulating this behavior is, olfactory transduction, and we're seeing upregulation of these genes when we switch from migratory to sedentary behavior. So what does this actually mean? This means that behavioral changes in brown bears that we're observing as a result of human resources actually has a very strong genetic basis. We're actually seeing the architecture of adaptation in these animals, and we do see signatures of selection, and we do see heavily influenced changes in transcription factors, which actually shows that this behavior can evolve. So what we're actually seeing is we're seeing evolution of these organisms to their new environments, to the human dominated landscapes, to the urban environments which they're seeing. And again, for me, that is a very important indicator that when we're trying to conserve these species, the question shouldn't be, okay, should we restrict them to these small forested areas, not allow them to use these human resources, or should we structure our conservation efforts knowing that these animals are adapting to their new environments, and we should actually start protecting them so that they can actually continue to adapt and continue to flourish in these new environments which they're forced to live in. Again, understanding which way evolution is going. So we actually don't know which type of variant, so whether sedentary or migratory behavior is increasing. So we're actually doing a study to answer that. So we're now collecting scat samples from all over Turkey. We've actually done this. So this study is mostly done by Mortaza Naderi, postdoc here at Koç University, and our master's and PhD students, Elif Çeltik and Ercan Sıkdokular. So this last summer, they did an incredible job. They went all over Turkey, collected over 700 scat samples from Turkey. And now we're in the process of sequencing these. And once we've sequenced them, we're going to look at both, overall genetic diversity patterns within Turkey of brown bears, but we're also going to look at, human versus environmental impact on adaptation. And we're also going to look at whether sedentary and migratory behavior, which behavioral type we're seeing more frequent and whether that frequency is increasing within Turkey. So that's going to be very important in telling us which type of lifestyle or what variant should we be protecting. We also have a proposal in place. We're actively seeking money for this. So other thing we want to do is we want to look at long-term evolution. This we're going to look at together with Matteo Fumagalli from Queen Mary University, together with Rana Özbal from the archaeology department, and Füsun at Hacettepe, who works in archaeogenomics. So what we actually want to do is we want to understand this dynamic from a long evolutionary perspective. So again, brown bears in general have been living together with humans for a very long time. So this interaction is very long. It's not new. Again, bears have been living in the same habitat as humans for a very long time. So we're guessing that this adaptation and this response to human landscapes has been emerging for a very long time. So what we actually want to do is we want to compare adaptation to changing environments in brown bears over the period of, for example, starting from somewhere around 400,000 years ago in the early Pleistocene, all the way to the beginning of the Holocene and end of the Ice Age to the beginning of the interglacial, and then how brown bears adapted to environments all the way up to somewhere around here, which is 7,000 years ago, where there was the agricultural revolution, and then all the way up to the industrial revolution. So what we actually want to do is we also have modern genomes to compare modern and ancient genomes to understand how adaptation occurred before industrialization, so how bears adapted to changing environments until the agricultural revolution, when there were no cities, and then after the agricultural revolution to the industrial revolution, then how that adaptation proceeds after the industrial revolution. So we're going to try to understand some of the modern patterns we're seeing here. When did they emerge, and to which responses, to which changes did they emerge? And is there a difference in how these animals adapt to environmental changes before and after human influences? So that's the major question. And again, we've collected hundreds of ancient bear samples, and this took like 2 years to finally get this collection, which we've collected from various dig sites in Turkey. And now we're actually waiting on funding that'll enable us to sequence all of these ancient genomes together with modern genomes to answer some of these very interesting questions. And again, this can inform us hugely about, like, if we understand where adaptation is going, this can really influence about how we should be conserving these species. Some other stuff that we're doing in the lab, it's all about understanding environmental change. So one thing we're interested in is environmental change is always, in the lab, it's studied very, what we're actually doing is it's studied in always fixed temperatures. What we actually want to do is, if environments are changing, if we have global warming, it's always stochastic change. So we want to actually see genomic responses to long-term stochastic change. So again, we are setting up experiments in different temperatures. We're using Daphnia magna as our model organism. What we actually want to understand is, how does adaptation proceed when temperatures increase in a stochastic nature? And what we actually want to do is we want to contrast between phenotypic plasticity, so quick adaptation through phenotypic plasticity, and how or whether that can switch to long-term genetic adaptation. So we're really looking at the genomic architecture of this, and this study is led by my postdoc here at Koç University, Nicola, and we just have a new master's student recruit, but we're also trying to recruit other students for the study as well. So those of you interested, we still have place for one more. The other is that together with Şenay Topaloğlu from TAGEN, we're also looking at the evolution or evolutionary origins and evolutionary genomics of lentil domestication. Again, lentils are, natural, or natural lentil is mostly found in Turkey or around the Fertile Crescent. There are other areas which we actually have natural lentil populations, but most of natural variation is still harbored in Turkey, and the rest of the world you can actually see only the commercial, the lentils that we eat. And what we actually want to do is we want to look at how domestication, the evolutionary genomics of the domestication of the species in general. And again, we're sequencing this data. Hopefully we'll get this data back in the next couple of months. And then again, we'll have a lot of place for bright students who want to analyze this data. And lastly, we're also trying to understand genetic architecture of adaptation to environmental gradients. So one thing we can do is, like, if we can't study evolution in time, we can study it in space as a proxy for time, and studying animals that actually can survive in lots of high, different gradients. So here we have a cricket species. We're interested in this cricket species because the same species can survive in 300 meters all the way to about 3,000 meters. So that's a huge environmental gradient where this animal can adapt to. And we actually see color variation, body size variation. There's lots of variation. And so what we actually want to do is we want to understand the genetic architecture, whether it's phenotypic plasticity or whether it's adaptive genetic variation that enables this organism to actually switch or continuously survive within this environmental gradient. And we're doing this work together with Hakan Gür from Ahiran University and Arda Cem from, again, Hacettepe University. And this project is, again, just newly funded. We do have place for one PhD student in this project. But again, those who are interested can always apply. These are all the funding agencies that have actually helped my research along the way. I thank every single one of them. And lastly, I'll leave you with the Ecology and Evolutionary Society, our society, which is going strong. We have a monthly bulletin. We also have every year we have a symposium. So those interested in evolution ecology, please, you can always attend. We have several different types of workshops, summer schools, winter schools for several different topics. Like each year we have one on evolutionary genomics. So if you're interested in this, feel free to apply. And we try to build a network as much as possible for those both young scientists and other scientists that are interested in ecology evolution in Turkey. And we have a wide network of researchers all over the globe, not just Turkish researchers here in Turkey, but also other researchers, other Turkish researchers, mainly all around the globe that work in these fields. So again, it's a very good chance for you to meet people. If you're interested in ecology and evolution, meet high quality researchers or researchers that do very good work all around the globe. With that, I thank you.